Perfect. All right, well, nice to meet everyone and thanks for joining this session. So I'm going to tell you a little bit about Domainex. I'm the Exec VP of Medicines Research here. So for those of you who are not aware of Domainex, we've been around for 20 years and we're an established integrated drug discovery CRO based in the Cambridge area. However, we have global reach. We work with biotech and pharma across the globe, as well as a number of academic institutions, really enabling their drug discovery process all the way from gene to candidate. And here is just a selection of some of the people that we've worked with and collaborated over the years. So during that period, we've really brought together a world-class team of over 90 scientists currently, but will be getting over 120 by early January. Majority of those scientists across the disciplines, which I'll come on to in a bit, are pretty much 70% have PhDs and postdocs with about an average experience in the drug discovery sector of around 8 years. And through this breadth of experience and that we've brought in, we've been able to deliver a number of clinical candidates for our clients, as well as have a number in preclinical development that's currently going through that pipeline. But we've also added value to a lot of our clients by generating novel IP, and we're inventors, or the scientists in the teams are inventors on over 150 patents and numerous scientific articles as well. So we've brought together a multidisciplinary team. So this goes all the way from chemistry, assay biology, and protein science. Now, we originally started as a protein science company, and we really like to try and utilize that in order to structurally enable our projects, but we can work in every single paradigm as well. So within the chemistry teams, we have really experienced medicinal chemists, synthetic chemists, analytical, bioanalytical teams, as well as computational teams, enabling that small molecule research primarily, all the way through to lead optimization and candidate selection, and working hand in hand with the assay biology team to create assays in order to set up screening, but also to really understand the physiology of the compounds. And we're also renowned for our biophysics platform that we've heavily invested in. So companies come to us in order for us to work on their programs all the way from gene to candidate selection in a really integrated fashion. However, a number of our clients we also work with aren't ready for that approach, or have got specific issues that they're tackling on their current projects internally. And we support them to do that with a number of different platforms that we use in an integrated fashion, but we can also use to unlock challenges that everybody is currently facing. So going from sort of the gene area, as I say, we're highly experienced in protein expression, whether it be in extracellular proteins, but also unlocking more challenging membrane proteins, and as well as in the structural biology, obtaining soluble protein in order to deliver crystal, in order to get crystal structures, and more recently Cryo-EM, and I'll come on to that in a little while. We also use screening teams in order to obtain novel compounds, in order to unlock targets, in order to get new small molecules. So we can do virtual screening, fragment screening, as well as covalent inhibitor screening as well. And this also goes alongside high throughput screening as well, which we can use to unlock different projects. And this is then really underpinned by the assay biology, making sure we've got key screening assays in place, but also looking at screening cascades and making sure we're looking at selectivity and various other paradigms as we move to sort of hit to lead optimization of those compounds, where we bring in those experienced chemistry teams and medchem teams in order to make sure we're optimizing compounds. And again, this is all underpinned by the other disciplines, including ADME and pharmacology. But the really emphasis here is that there's no one solution that fits everybody. So we're highly collaborative and work to customize our science in order to make sure it fits in order to unlock your targets of interest. So once we've got small molecule hits, whether it's from our internal platforms or from our clients, we then look to take this through all the way through the drug discovery process. And we're with you every step of the way whilst we do that. We've got experienced people who have worked in the industry for over 30 years, where we're looking at establishing the target product profiles of those small molecule compounds and then continually reviewing them to make sure that they are fit for purpose as we go through the more expensive end of animal model studies and also scale up in order to go to IND ready status. And in order to do that, we work with a number of strategic partners who we've worked with for over a decade, and to name a couple here, Crescit and Pharmadex, which again, both local to the Cambridge area. So once we've got those hits and they're validated, we really encourage and really express with all our scientists in the medchem area that it's not just about optimizing one factor when it comes to, for instance, potency of compounds. And we really look at the analogy of the Rubik's Cube, where everybody can solve generally one face of that cube, in this case, potency. But in order to develop and optimize a compound of interest, there are a number of different parameters which you must understand, and each of them impact the other. So in order to solve that Rubik's Cube, you've got to understand and make sure you're keeping an eye on all of those different criteria. And we really work in a multi-parametric optimization fashion, where we emphasize that every compound that we design is important and work on the basis of every compound counts. And this then really has enabled us to generate high quality compounds for our clients. But it's also about speed and efficiency, and it's going around that design, make, test cascade as quick and as efficient as possible. And we've built up our teams in order to give them all the required software and computational tools and experience they need to do the design, go through to the synthesis of those compounds and generate those compounds in as quick as possible, to go through to screening, when we've delivered data in an average 5-day turnaround time, depending on the assay, and whether that's in biological output or also in the ADME, in order to get that data, to do the analysis. And again, we have all the databases and facilities in order to make sure we can generate and do that analysis. And for us, as I said, it's all about collaboration, making sure that we're talking through these results, making sure our clients are understanding them, and working in collaboration to generate the best compounds as possible. And we go through that design, make, test paradigm roughly about 5 times in a 3-month process. And so we can make all those key decisions hand in hand with our clients. And as I say, then we've worked on this in a number of areas. We've got a proven track record of success, but we've also invested in a number of different solutions that are enabling timely and cost-effective solutions for our clients. Our teams are highly scientific driven, and going on to that customizable nature, everything is tailored for you and your projects. And again, we've invested in a range of technologies in order to make that happen. So a couple of areas that are obviously in vogue, I just want to highlight them at the moment. So protein degradation. So we've worked in the PROTAC drug discovery area for a number of years. We've taken compounds that we've prepared up to multiple gram quantities, enabling PK and animal work. So we've got all the experience to do this. We've worked on known E3 ligases, but also worked in targeting novel E3 ligases that our clients have identified, or again, identifying through virtual screening or fragment screening, novel ligands in order to target the current E3 ligases. And we've set up a whole platform in order to understand the degradation of proteins, but also understand the binary and ternary complex formation of those compounds. And again, bespoke to Domainex, that innovation created cell accumulation assays where we can look at how much of those PROTACs have got into the cells of interest, again, so we can optimize the correct chemotype and make sure we're working on the right compounds going forward. And then mentioned earlier in the Cryo-EM space, with an established protein science company of origin. We always generate high quality protein, one of the key factors, and we've also been able to attract a number of key structural biologists with cryo-EM experience. We're based in Cambridge. We have access to the state-of-the-art facilities with a number of microscopes, and through that, we're now generating cryo-EM maps in order to unlock targets and work on structurally based drug discovery programs that we can start to interrogate ligands in order to pass that through that process to generate those clinical candidates. So it just needs me to start wrapping up. So we've put together a talented, experienced, multidisciplinary teams. We've got cutting edge technologies to unlock those challenging proteins, and we work in a highly collaborative manner. This has enabled us to generate high quality, cost effective and timely data driven projects that's really customizable to your needs, generating value by unlocking novel IP and taking your compounds all the way through to the clinic. And then I'll just finish off with a slide just so you can have a look in your leisure relating to the website. There's various links there, but I'll just highlight a couple of press releases we've had more recently where we're working with Parkinson's and NRG in the neuroinflammation space for Parkinson's research, but also in the oncology area where we've unlocked a challenging, unthought of, undruggable target protein-protein interaction of the FLIP inhibitor program, where it enabled Queen's University Belfast to go through a licensing agreement with Ipsen. And so, yeah, if you have any questions, either reach out to me or I'm more than happy to answer any now. So, and thank you for all your attention, but also for the Domainex team in order to be able to put all this in place in order for us to work in that drug discovery area.